Antibiotic resistance. Let's review the mechanism of penicillin. Penicillin's target is a transpeptidase enzyme. And old penicillin has this four-membered ring of a beta-lactam ring. Beta-lactam is a chemically unstable structure. So that's why when it binds to the enzyme, then this four-membered ring is broken, and then it can attach to the transpeptidase enzyme covalently, and it can kill the enzyme function forever. So that's why this beta-lactam ring is a key functional group of all the penicillins. Bacteria's strategy is the breaking up this beta-lactam structure utilizing beta-lactamase neoenzyme. So beta-lactamase is evolved, and if this enzyme break up the penicillin, then the bacteria can survive. So this is very obvious and first case of the bacterial resistance to penicillin. Another possible mechanism is changing the structure of a target protein. Now, it was discovered from methicillin-resistant strains. Methicillin is one kind of improved the penicillin structure, and the target of methicillin should be the transpeptidase, but that enzyme structure was mutated and changed, and it does not bind to the beta-lactam anymore. That's why they can avoid the attack by the penicillin. So this is the second mechanism of the antibiotic resistance. Third mechanism, simply if the bacteria take up the drugs, they pump out. So they develop this efflux pump more efficiently. Any kind of a toxic material inside, they just remove from the bacteria. So this can be this is not only for the penicillin, it can be general mechanism to fight against the antibiotic attack. Another mechanism is similar, but even before the drug enters the bacteria, they block those transport from outside to the inside. So by changing those transporter protein on the membrane, they prevent from the beginning, do not allow the access of any the penicillin molecule into the bacteria. So this is fourth mechanism. Once this kind of the resistance is achieved by one bacteria, those information can be easily shared by other bacteria. So bacteria has a genome DNA, but at the same time it has a smaller the plasmid, circular plasmid, and this plasmid can be shared by other bacteria. So one bacteria's ability of the antibiotic resistance can be shared by other bacteria. Very interestingly, this sharing is not only between same bacteria. This plasmid can be transferred to totally different strains of bacteria. So this makes the whole situation of bacterial resistance is much worse. So this is the summary of various antibiotic resistance mechanisms.